Royal Institution Christmas Lectures, Lecture 6, the last lecture. "20 seconds. Ten, 9, 8, 7, 6, 5, 4, 3.
I hope that I've not by now given you the impression that we know all there is to know about living things and the molecules that we're made of, but I hope too that I've given you the impression that we know something about it and that it's quite exciting knowledge. But is it useful knowledge? Can we actually get these molecules to do anything for us? Can we put them to work? Well, human beings, of course, have been doing that for thousands of years. Since the earliest times, men, without knowing it, have been using enzymes, and the result of that is that quite a lot of the food that we eat, especially, oddly enough, some of the things that some people enjoy most, are produced by enzymes for us. And of these, perhaps the earliest was concerned with making cheese. Our early ancestors, keeping their herds, moving them from place to place, they perhaps carried milk in the stomachs of dead animals, and they found when they did that that something funny happened to it because there was in the stomach an enzyme. So let's have a look at that, Bill. Here's some warm milk, which Bill is putting in a flask. And he's going to add some enzyme to that, rennin. This is the stuff from the calves' stomachs. And if he stirs it up and we wait a little bit, we hope that something will happen if the temperature was right.
It's starting to happen.
It's starting to happen. Yes, the milk is thickening. You'll have seen things like this if you make junket. And something is separating out. So we are getting now, yes, I can see that. What we are getting is curds and whey. So if Bill pours that into a sieve, you'll see that a fairly cloudy liquid comes through, not as cloudy as the milk was originally, and left in the filter we have some protein which is precipitated. Now what's happened is that the major protein in milk, casein, has been split by the enzyme rennin, and that's changed its properties. It's coagulated, become insoluble, and there are the curds which could then be pressed and left in appropriate ways to produce cheeses. And over there we have the liquid which has gone through, the whey. Now this still contains some protein, but in addition to protein, it contains some sugar. And that ought to be useful, some sugar. But I haven't talked very much about sugar. What are sugars? We've encountered them. They were present in the bacterial cell walls, and I called some things sugars in the nucleic acid structures. But what are sugars? In one sense, they are even more fundamental than the proteins and the nucleic acids we've been talking about, because the sugars are the molecules that are made in the first place using energy from the sun. Using the sun's energy and carbon dioxide and water, these molecules get split and joined together in different ways so that 6 of each get added together to produce a molecule of a sugar, glucose, as shown in this model, in which there are 6 carbons, 6 oxygens, and 12 hydrogen atoms, and in addition, oxygen is released in the process. And here you see that the molecule consists of a 6-membered ring where one of the atoms in the ring is oxygen and the other 5 are carbon, and all of the carbons are attached to 4 other groups, so there are oxygens spread out around the outside as well. It's a rather complicated looking molecule, and it can in fact take a number of different shapes. Perhaps that may be clearer if we look at it in a diagram. I've tried to draw one of the possible shapes of the molecule. This is glucose in the shape which we call a chair shape. This line is thick because that's out towards you. And you see that there are the carbon atoms and here are the oxygens and hydrogens attached all round. That's a glucose molecule. Now these molecules get joined together end to end to form polymers. And the most common organic material in the whole world is the stuff of which trees are made, cellulose in the main. And we can see that in the next drawing, in which I've tried to put together 2 molecules of glucose. Here's one the way around that we were just looking at one. That's 2 molecules of glucose joined together through this oxygen in a particular way. The carbon of this glucose to this oxygen to the carbon of the next glucose, and so on down a very long chain. That's cellulose. Cotton is about 100% this material. But why don't we eat this stuff? The fact is we don't have enzymes that can break it down. Most of the enzymes, and indeed those we're going to talk about today, are concerned with manipulating chemical reactions involving sugars. But we, human beings, don't have enzymes which will break down cellulose, which is why it's no use eating grass and expecting to be nourished by it. Even cows don't have this enzyme. They get them from bacteria who live with the cows, as we'll discuss in a minute or two if I remember. But the stuff that we do eat, starch from potatoes and wheat and so on, that's also made of glucose molecules, but joined together in a slightly different way. If I detach this, and instead of joining the carbon to the oxygen in the way that it's done, I use the chemical bond from the carbon which was previously to hydrogen, turning it round like that, then I get a new sort of linkage, and that's the sort of linkage which is mainly present in starch. And we do have the enzymes to deal with that, which is why we can break down starch molecules into separate glucose molecules, and then we have enzymes which go on beyond that, extracting the energy from the food. You can see in this diagram, if I put it back the way it was, that these sugars, which are fascinating chemical molecules, provide all sorts of possibilities for doing things differently. Suppose, for example, that I pick on this oxygen atom. No, I won't pick on that one. Let's pick on this oxygen atom connected to that carbon. I could connect it differently. I could have connected that one perhaps up here where the hydrogen is. That would turn into a slightly different sugar. Well, let's look at a slightly different sugar. Here's a drawing of another sugar made of 2 units put together. There's another sugar made of 2 units put together. Looking at that carefully, can anybody spot a difference between the two? Any offers? Yes, the girl at the end.
That thing's bended up.
The one, which? The one over at the left as you look at it?
Yes.
Right? Well, that's right. Look, this oxygen is attached on that bond, whereas down there it's attached on that bond. So that's the only difference between this one, which is 2-glucose, and this, which is something else. In fact, we have a name for this sugar connected differently. That's galactose. So this is galactose attached to glucose, and that's actually lactose. That's the milk sugar, the stuff that's in the whey. That doesn't taste quite so sweet as glucose, and it's not as soluble, so that's not as convenient. And furthermore, although we think milk is a very good food, quite a great proportion, perhaps as much as 50% of the world's population, isn't capable of digesting lactose properly. And that may be an enzyme deficiency. They don't have the enzyme that will take it to pieces. So if we are going to use milk for food for lots and lots of people, it might be better to take the lactose to pieces first and have galactose and glucose. Let's see if we can do that. What we would do, if we start bringing out the apparatus, is add the lactose to some enzyme, and the enzyme, a specific enzyme, which has to recognize this particular sugar, would cut it in half, and then we'd have the two pieces, and off they would go. But unless we're very careful, the enzyme will go with them as well. We'll have to be more clever about it than that. How shall we handle that? Well, here's an apparatus brought over specially from France, where the experiments are going on, in which the Milk Marketing Board is really very interested, which shows how it might be done. What we need to do is immobilize the enzyme somehow. So in this apparatus, here is a little bed in a column here, a little bed of glass beads. And the glass beads have the enzyme attached to them. So if the lactose is coming out of somewhere, is that coming out of this one? The lactose comes in at the top, down the column, over the glass beads. The enzyme works, and out of the bottom comes a mixture of galactose and glucose. Now that ought to taste sweeter. Let's see if we can discover that. Let's have some volunteers. I want a couple of people to test. Yes, that girl sitting on the end of the row looks very good, and the boy behind her, I think. Splendid. Now, let's see if we can find out whether this is sweeter. Now, I think you mustn't look, really, must you? So you need to stand over there, and could you face over in that direction so you're not looking? All right? Something like that. Now, meanwhile, this is the one coming in, and that's the one going out, and I'm going to put labels on those so that you'll know what I've done. Here's the one coming in, the lactose, and there's the one going out, the mixture, and I'm not telling them what letters I've put on it. All right? Now then, if you like to turn round, we are all set with these interesting mixes. I've lost my label, but never mind. Let's give you this one first. So turn round a little bit. This way. Now, open wide. What I want you to remember is how sweet this was. Right? You're getting liquid B at the moment. How sweet is that going to be? And I want you to remember, too, how sweet is this? All right? Just hold on to that recollection. How sweet is it? Here's liquid A that's lost its label. Now, remembering how sweet the first one was, see if you can detect a difference. Just savor that for a moment. Let's see how sweet that is. Here you are, last dose coming. Open wide there. Now then, can you tell me which of those was the sweeter? That's the last one that you just had. Well, that's very good, you see, because that's the galactose-glucose mixture, and the first was the lactose mixture. Well, good. So immobilizing the enzyme like that, we can have a commercial process which will break up the lactose into galactose and glucose, which has all sorts of advantages. That's not the only sort of complication that we get in sugars. Let's have a quick look at this diagram, which is drawn in a more conventional, stylized sort of way. We've got glucose again over on this side, on the left, drawn in this conventional way, but over on the right we have a different arrangement, though it includes exactly the same number of atoms, based on a 5-membered ring. Now this is fructose, a different sugar, which is a bit sweeter than glucose. So there are enzymes which will change glucose into fructose. And if we wanted to do that, we'd adopt a similar sort of process. Here's the apparatus that would do that. This time there's a much longer column of enzyme here, and it's been produced in a different way. Not glass beads this time. Now the enzyme molecules are linked one to another, millions and millions and millions of them, making up this column. And if we flow glucose in at the top, what comes out at the bottom is a mixture, half and half, glucose and fructose, which we might want to achieve. That's another important commercial process for transforming, say, corn syrup into this mixture of glucose and fructose. Glucose and fructose actually come together, joined as you see in this diagram, in the commonest sugar of all, the table sugar that we're all accustomed to, sucrose. In sucrose, that's two sugars, glucose, fructose, joined in this particular way, and of course there are enzymes to handle that. I hope you're getting the impression that these subtle changes in positions of atoms in the sugars all demand that the enzyme should have slightly different shapes to fit them to handle these particular chemical reactions. And furthermore, you should be noting that these sugars are rather difficult to handle chemically. You see there are all these oxygen-hydrogen groups sticking out all over them in rather similar environments. So if you're doing chemistry in a test tube, it's difficult to react with one and not the other. But an enzyme can detect the exact shapes and pick the reactions it really wants to promote. Now there's sucrose then, the really common sugar. Have you ever wondered how liquid center chocolates get their liquid centers? Let's have a look at a liquid center chocolate. I've got some chocolates and we'll be celebrating the end of these lectures afterwards with a chocolate or two. Let's see if we can find a liquid center chocolate. Yes, here we are. I think this is one. Let me waste this one by cutting it open. Ah, look at that. There you are. There's a lovely, gooey, runny center to that chocolate. How did that get inside? Any suggestions? Was it injected? That would be very labor intensive, I think. A lot of people would have to work in the chocolate factories to do that.
Half the chocolate made, it filled up, and then the rest of the chocolate put over the top.
Well, that's good. You make a cup of chocolate and pour in the liquid and put the lid on. That would be fairly difficult to automate too. No, we are talking about enzymes, and you really ought to be looking for an enzyme solution, and I think we have to go on, and I'll tell you at this point. This sugar, sucrose, is not very soluble in water. And you know that from making icings to cakes and so on. You can make a nice icing as a paste, put it on the top of a cake. So if you make a paste with sucrose using not very much water, then you can put a chocolate coating on that easily enough. But suppose you put in an enzyme as well, which divides the sucrose into glucose and fructose. They are more soluble, so they dissolve in the water that was there that only made a paste with the sucrose. Now here, by courtesy of the manufacturers, I have a splendid supply of chocolates. This one has got a paste but without the enzyme in it. Right, and if I just do that, that just breaks, and there's the pasty center. Here's one that has the enzyme in it, otherwise just the same chocolate. If I try to break this in the same way, that doesn't break so easily, and I can pull it out, and there's a nice gooey center, look. So there's an example of enzyme technology, and we'll hope that your mouths can water about that until the end of the lecture. Then something might be possible. Now these reactions we've been talking about simply involve one enzyme. That's not very much, just one enzyme. Suppose you wanted to do something rather more complicated, again with sugar, a more complicated set of reactions, to make something which was more than one step away. Then you'd need a lot of enzymes, and it would be rather troublesome to put them all on glass beads and all the rest of it. What would we do then? Well, why don't we use a nice friendly little bacterium or a nice friendly little fungal cell, a single-celled organism that had all these enzymes inside and which would do what we wanted? Well, people have been doing that for years and years and years. That's how bread is made, say. I don't know what I can do with this. Can I cut this? If you... Very good. If I try cutting this piece of bread, like that, I don't know that it shows it very well because it's a bit fresh, but you'll know what bread looks like. It has a lovely porous sort of structure, full of air bubbles. And you'll also know that bread is produced using baker's yeast. Here's some yeast. A nice fungus is yeast. Single cells, bags of enzyme. And what the enzymes are doing is shown in this little diagram. The enzyme is working on the glucose in the dough. There's the glucose, and it's breaking it down through many steps of chemical reactions to produce these molecules over here. Those at the bottom are 2 molecules of carbon dioxide. That's a gas. There are the gas bubbles from 1 molecule of glucose. And these 2 rather odd-looking little molecules, which are these toxic things which people are so fond of. They're alcohol molecules. Didn't know there was alcohol in bread, did you? Well, that mostly gets driven off, but it contributes a lot to the taste of baked bread. Now, bread isn't the only thing that's produced that way, of course. Wine and beer are produced that way. Here is Bill's home wine factory with the grape juice and the yeast, slightly different yeast in there. And here's the airlock at the top, and you can watch bubbles of carbon dioxide coming off there slowly, while the glucose inside is being broken down and alcohol is being formed. Now, as I said, that takes a lot of reactions, something like 15, each one catalyzed by an enzyme. So let's break off for just a moment and look at one of them so you see the degree of detail that's involved. This is just one of the reactions illustrated here. It's important at one stage in these reactions to change this molecule into this one. And no doubt you could spot, if I left you a minute or two, what the difference is. The difference is that in this one on the left, there are 2 hydrogens connected to this carbon atom. There's one coming out of the diagram towards you, that one. Whereas over on this side, there's only 1 hydrogen connected there, but there's a hydrogen connected to the carbon atom next below, just there, and that's coming out towards you. This enzyme has to move a hydrogen atom from that position and put it in at that position to produce that molecule. That's the kind of detail in which these enzymes work. And since this is one we've been working on in Oxford, and which I think, from my prejudiced point of view, is rather pretty, let's have a look at it. Here's a model of that particular enzyme, and we've arranged it so that I can light it up. And what I'm going to show you is that it's made of little units, some of which are like silk and some of which are like hair. In some of them, the protein chain is drawn out, and in others, it's in a helical form. So if I switch on the switch and we start on these, here we have some lights coming up on the part that's like silk. And I hope you can see, and you could see even better if I turned it around a little bit, that they're forming a sort of barrel-like structure in the middle of the molecule. Now if I switch on the other lights, these are the bits of hair, and they're arranged around the outside of that barrel made of silk. There we are. So we have the silk in the middle and the hair around the outside, making, as I say, what I think is rather a pretty molecule. It's quite big. It has thousands of atoms in it, And since Christmas is nearly over, I'd better switch it out and go back to say something else about it. All those atoms, and it's going to bring about this rather detailed chemical reaction, you could probably guess the sort of way in which it does that. It would need to have a good negative charge over here, pulling at this hydrogen atom, pulling off the proton, and a positive charge over there, pulling the electrons out of these bonds over in that direction. And then it would need to go the other way around, deliver the proton there, and let the electrons come back to make a bond there. We all must understand, I hope, how that's working, and we understand that for most of the enzymes involved in going from glucose to alcohol and carbon dioxide. Well, there we have some rather useful, friendly bacteria, or at least a friendly fungus. We did an experiment in one of the earlier lectures on bacteria. We wondered whether we could actually grow some, and here are the results of that experiment. Are any of the people who helped me with that still here? Yes. Now, one of you was brave enough to kiss one of these plates. Do you feel brave enough to come out and see what happened? And we'll start by having a look at not the kiss, perhaps, but at, that's the kiss. Let's look at a thumbprint, and we'll see whether there's anything in that. Yes, I think the best thumbprint was this one. Now, what you'll remember is that thumbs put on, and this is a nutrient material in here, so we'd expect bacteria to grow if there were some present on the thumb. So what we see on the one with one thumbprint on is a rather lurid impression here, showing that great colonies of bacteria have grown. Some of them are nice single little patches which might have developed from just one bacterium, but you'll agree that quite a lot have grown there. Now, what about that brave kiss? I was hearing on Radio 2 the other day that What's in a Kiss was the most popular song on Radio 2 last year, and now we'll begin to find out. This is what we see. Here's a nice little patch in the middle there. That's the mark of your lips. And just above that is another little patch. That's where the end of your nose touched it. So you see, you were going around with some bacteria, but don't be worried about it. We all are. Thank you very much indeed. That was brilliant. Now we can cope with those bacteria, perhaps, because the skin is not easy for the bacteria to get through, and that defends us. If they get into our mouths and we swallow them, then the juices in our stomach can destroy them. And if they get into our eyes, as we saw last time, we have some lysozyme, and that can destroy many of them. So many of these bacteria are not very harmful, but some are harmful. And we get sore throats, and it's possible to get blood poisoning and so on. What can we do about those? Well, that brings us back to Fleming again. You remember we talked about Fleming with lysozyme. Fleming did an experiment about 10 years after that of a rather similar kind. He left a bacterial culture on his laboratory bench with the cover off. And while he was away, again, a fungus came in and colonized it and grew on the culture. And when he came back, Fleming found that where the fungi had grown, there were patches where the bacteria were dissolved away. That's what it seemed like to him. So he thought, goodness, the fungus is producing something which will kill the bacteria. It doesn't work quite like that, as we shall see, but it was very exciting, and he tried quite hard to isolate the material. He called it penicillin because it came from the penicillium mold. But he didn't succeed in isolating what he'd called penicillin. He didn't know what sort of molecule it was, and that waited until Florian Chain in Oxford, about 10 years later again, after a very intensive effort, did succeed in isolating it. And I can show you here the chemical formula of that. This is the chemical formula of penicillin, which is, by comparison with the things we've been looking at, rather a complicated molecule. But it's very effective in killing these bacteria, as we know. How does it do that? Well, let's go back and have a look at the drawing we saw earlier of the bacterial cell wall. Here's the drawing. And we looked at this when we were talking about lysozyme, because it's an interesting fact that both of these discoveries of Fleming's relate to this one drawing. Here are the chains of sugar molecules in the bacterial cell wall, and that's where they are punctured by lysozyme. But these cross-connecting pieces, which are a bit like protein, these have to be built up by the bacterium using enzymes. And somehow penicillin affects the way in which these cross-connections are built up. We can see, in principle, how that might happen. Let's go back to our enzyme model. Here's our jigsaw puzzle enzyme model again. And you'll remember, and let's suppose this time we relate it to the bacterium building up its cell wall. Along comes some cell wall components. They fit onto the enzyme in an exact way, and they are linked together, and there's a new connection making the cell wall, and that goes off again, and the enzyme works again. But suppose there is in the environment a molecule which fits part of this and has nothing to do with making the bacterial cell wall. It just fits onto the enzyme and sticks there. Then when the real components come along, they can't get in at the enzyme, so they're not joined together. This molecule is preventing the making of these connections. Well, it's thought that that's how penicillin works. It sticks onto an enzyme involved in putting in these cross connections and prevents them being made properly. So instead of a bacterium being made, which has this nice robust wall and is all very stable and goes about doing no good, the bacteria, as they try to grow, can't manage it very well. It's like perhaps blowing up this balloon. Let's find somebody with a good pair of, here you are, why don't you have a blow at that? And then we'll see a bacterium trying to grow and not quite managing it because all the bits are defective. Yes? It's not quite manageable, is it? So it's not that penicillin probably kills existing good bacteria, it prevents them growing in this way and is extremely effective. That's not the only sort of molecule which is useful as an antibiotic, though it remains one of the very best. Let's think about another one. In the slide, we have another possible antibiotic, and this is a rather complicated looking formula. You see it has this planar group down here connected to all these atoms, which are closely related to a bit of protein chain. That's a sort of flat chemistry diagram, and they don't tell us all that much. What does it look like in 3 dimensions is the question we're beginning to ask ourselves, isn't it? And this is a model in the space-filling components of the antibiotic actinomycin. And here's that flat planar piece, and round here are the bits of protein-like chain. How do you think that might work? Well, let's have a clue, Bill, shall we? Here's our double helical DNA. This molecule can come along to double helical DNA, force its way between adjacent base pairs, and fit in there really rather neatly, with this part specifically fitting on to the rest of the helix and stopping it being pulled apart. So it can't be pulled apart for the RNA to be made on it. Now, there's a problem with that. The bacteria that we want to kill or the cells in our own body that we want to kill would have DNA, but also the cells we don't want to kill would have DNA. So it's not like penicillin. The great thing about penicillin is that only the bacterium is interested in making that cell wall. We are not interested in our bodies in making cell walls like that. So we don't have those enzymes, and penicillin, it doesn't matter, can't do anything to us. It can only do things to the bacterium. So it's very selective. This is useful, but has to be handled much more carefully because it's not so selective. Now, so much for bacteria. Bacteria aren't the only things that are nasty. There are other nasty things about. What about viruses? Let's have a look at a model of a virus. Here's a model of a virus. It looks remarkably simple, doesn't it? It's a lot of red balls, look, making a shell, and they represent protein molecules. They may be all the same protein type of protein molecule. Each of them, shall we say, as big or bigger than a lysozyme molecule. All folded up to form a closed spherical shell, and inside there is nucleic acid. So this is rather a stable thing. You couldn't really call it alive. Protein on the outside, nucleic acid on the inside. And what a virus has to do in order to get more viruses is to find a cell that it can infect. The nucleic acid then gets inside the cell. It takes over the cell's machinery to make more virus, nucleic acid and virus protein. The cell dies, out come lots more little virus particles, which at that stage are just protein nucleic acid complexes. Let's look at a slide, and you will see there some poliovirus inside a cell. And these spherical particles have all packed together in this typical six-sided figure honeycomb sort of structure. They've crystallized inside that cell. Now that honeycomb structure, you have to remember we're talking now of different levels of structure. The honeycomb structure suggests a way in which spherical things would pack together. There they were spherical viruses, but what about these spherical protein particles of which the virus is made? We talked last time about self-assembly. How does a spherical shell assemble itself? Well, you'll know that if you have a lot of balls and you collect them on a flat sheet, what they're likely to do is to pack nicely together, as you saw the virus particles inside the cell doing in this regular array. But that's all flat. How do you turn that into a spherical surface? Well, Aaron Klug and Don Casper had a good idea about that. They said, let's take out a slice, a segment, as has been taken out in this little sheet. Then it could fold up to form a sort of pyramid instead of forming a flat sheet, except that now in the middle of that, at the peak of that pyramid, there would be a 5-membered ring with one in the middle instead of a 6-membered ring. But the contacts between the balls would be very much as they were in the flat figure. So to make a spherical shell from that flat figure, what you have to do is make some of the 6-membered figures into 5-sided figures. Well, you all know that. That's how footballs are often made. Here's a football. There, in this position, is a 5-sided figure, and around it we have these hexagons, turning this into a nice spherical shell like a spherical or icosahedral virus. So how do we deal with viruses or with bacteria if the antibiotics aren't any good? Well, here we have some more protein molecules in our bodies, antibodies. Let's have a look at the structure of one of these antibodies. This is a protein made up of protein chains of 2 sorts, and this is just a formal diagram of the arrangement. Here's the end of one chain painted black, and that goes down, and I've looped it round like that because, again, the detailed structure is rather the way it is in silk, and it goes on down to there. The other black one starts there in the same way and goes down to there. And there are two shorter chains, coloured red, which fit on there and there, like that. Incidentally, the green bits in the bottom are sugar molecules attached to the protein. Now, in this Y-shaped molecule, then, there's a little patch there, which is an exact jigsaw puzzle fit to a foreign molecule. And over on the other side, there's another patch, which is an exact jigsaw puzzle fit to a foreign molecule. So if a foreign molecule, like a virus, gets into your blood, these antibodies come along and make it clump together. Let's have a little experiment to do that. What I want fairly quickly is 3 boys to come out. Let's have the 3 at the end of that line is the way to get you out. Come on, quickly. And then we'll have 3 girls to act, and the 3 in the front row will do very well, to act as... Now you wait back there just a moment, the girls. Would the boys put their hands in their pockets? Your viruses, and you're looking around for a cell to infect. Now, this is quite a good model of an antibody molecule. Here's one site to find a foreign molecule, and here's another site to find a foreign molecule. So if the girls come out with their hands stretched out, and with each hand they look for separate foreign molecules, we'll see what happens, right? Now, what you do is grab a virus with each hand. Now, the viruses aren't allowed to try to get away. Right? So now we are forming a clump. The viruses are all clumped together, and they can be eliminated by the body. They've been rendered immobile by these antibodies. Thank you very much. Now, of course, it isn't just viruses that are foreign. Foreign molecules can come about in various ways. We are not all identical, after all, and a thing that we know is that we have different sorts of blood. Now, we have different sorts of blood because the red blood cells carry on their surfaces, here's a red blood cell, carry on their surfaces little groups of sugar molecules, again, of different kinds. This one is carrying a kind called A, and this one is carrying some sugar molecules called B. And other cells have both sorts, so they're AB. And others, again, have none of these. They're neither A nor B. Now, suppose somebody with blood of variety B needs some blood. They need a blood transfusion. So we put in, by mistake, blood of variety A. Our individual B will raise antibodies to blood of variety A, and those red blood cells will all clump together, and that will get into a terrible state. It will be worse. It will be better not to have done it at all. So it's important to know the type of blood, and that's why people who give blood to the blood transfusion service, why they have to have their blood typed. Here's a National Blood Transfusion Service registration card from a person in the audience, and you can see that in the card, just up there, it says that his blood is of type A. When most of you are a bit older, I hope that you will join this organization and occasionally give some of your blood. That's really rather important. But how would it be typed? Well, let's see if we can do an experiment of that sort. What we need to have is a solution with antibody of type A in it. That would be antibody against type A. And another solution with antibody against type B. Then if we've got type A there, the red blood cells will be clumped together. All right, now, I had to get the volunteer earlier because we have to get permission for this kind of thing, but here comes Julia, the volunteer, and Bill is going to mop her finger carefully with disinfectant, and then he's going to use this clever new device to prick the end of her finger. And then, on a slide marked A and B, we're going to press out some blood and make 2 drops of blood there. Here comes the blood. Is that all right, Julie?
Yeah.
Put some on there.
Right. There's a spot of blood. And
Not too much.
are you bleeding well?
Yeah, she's bleeding very well.
There's another spot of blood. All right. Let's hope that's enough.
I think it'll be enough.
And now we'll put it on the overhead projector and hope to be able to see it there. There are the 2 spots of blood. And now if Bill adds to one of these spots some antibody to type A, and to the other spot of blood some antibody to type B, and then we mix that up. Now that's type B being mixed up. Is anything happening there? That seems still to be nicely mixed up, doesn't it? Right? Now let's mix up the A. Oh, now there, that's looking to me a little granular. Is that right?
Yes, that's great. It's gone.
So there the red blood cells are clumping together. Thank you, Julia. That means that putting in the antibody to type A has clumped those red blood cells together, so Julia's blood, we now know, is of type A. I think we should thank her for going through that ordeal. Bacteria and viruses. Funny thing is that even bacteria suffer from viruses. It's like this business of big fleas have little fleas upon their backs to bite them, and little fleas have lesser fleas, and so ad infinitum. Now this is getting rather close to the ad infinitum. If we look at this slide now, there you see the outline of a bacterium surrounded by all these bodies which are viruses. Viruses which prey specifically on this type of bacteria. If we look at the next slide, we'll see one in a bit more detail. Here you see, pointed at by the arrow on the slide, that has rather a splendid structure. And perhaps we could have a look at that. Let's have a look at a model of this viral structure. If I take it off, it's called a bacteriophage. It really is a rather splendid thing. Here's the head, looking rather like a spherical virus, made of protein in the same way, full of nucleic acid. And here's all this other apparatus, all made of protein. Quite a complicated thing. It looks as though it is almost alive. When it approaches a bacterium, let's say this is the beast, it touches down like a moon rocket, and then finds a spot on the bacterium to make a hole in it, using lysozyme, of course, to make the hole. It has its own lysozyme, and then in it goes and squirts in the nucleic acid, which then takes over the bacterium. If we look at the next slide, we can see an exploded view of such a bacterial virus. The protein coat thing is in the middle, and spread around is this long, long chain of nucleic acid, carrying the coding needed for the proteins of which that virus is made, and any other enzymes that might be necessary to assemble it. That suggests something really quite interesting. I'm not saying that this is actually the train of thought that happened, but could we use such a virus to introduce new DNA into a bacterium that we wanted? Could we, in fact, use a bacterium to make proteins that we want to make? Well, that's what genetic engineering is all about. What we would want to do, in principle, would be to take the bacterial DNA. So let's find a piece of bacterial DNA. Here we have it, a double helical piece of bacterial DNA, which is actually circular, so it will be folded around like this into a circle, if I can get it under control. There we are, a piece of double helical DNA. Suppose we can take that out of the cell, and we want to splice in a piece of DNA which will code for the protein we want to make. How do we do that? We use an enzyme. So let me find an enzyme. I've lost my enzyme. I better use this knife, I think. With my enzyme, I cut this piece of DNA there, and I can cut it again there. This enzyme cuts it in a very specific way, leaving these two sticky ends. Now if I make a new piece of DNA to code for the protein that I want to make, let's suppose that's this, which has the complementary sticky ends, then I should be able to splice this in, making my old familiar AT pairs over there and my old familiar AT and TA pairs over here. All right? So now I've spliced in my new gene for the protein that I want to make. What protein is that going to be? Well, it could, for example, be a protein that we're very concerned about. It could be insulin. What's insulin? Insulin's a protein. This is a substance which somehow controls the way in which glucose is taken up into living cells. If the insulin isn't there, then glucose can't get in the cells. The cells are not nourished, don't get the energy from the glucose, and that's bad. This is the unhappy condition which diabetics suffer from. So they have to take extra insulin, and that enables the glucose to be taken up. We don't yet understand quite how, but it does. So we'll come back to that in a moment. Let's look at the insulin molecule. Here's the structure of the protein. It's quite a small protein molecule. This is the structure worked out by Dorothy Hodgkin and her colleagues, and you see the insulin molecule folded up in this characteristic way. There's a world shortage of insulin. It would be very nice to find a way of making bacteria make it, and this is a possible way which is being explored. Then there would be enough, perhaps, to treat all the people who suffer from diabetes worldwide. What they would have to do, and what they do increasingly in this and other fortunate countries, is to be able to check the glucose levels in their blood so that they know whether they need supplies of insulin or not. And they do that with a rather clever little new system, a test kit. Here's a test kit. And if I take out one of these test papers, here's a test paper. They put a spot of blood on the test paper at this end here, and the paper then changes color according to how much glucose there is in the blood. How does it do that? Enzymes, of course. The paper has two enzymes in it. One of them produces hydrogen peroxide from the glucose, and the other one breaks down the hydrogen peroxide in a way that we've already seen to produce oxygen. And the oxygen interacts with the indicator paper to produce the color. I think we should start this on a larger scale so that you see the sort of thing that's involved. Here's a larger scale thing, right? Let's put the sugar on that. Here's a sugar solution, and we'll see a color change. That would be a spot of blood if we were using the test paper. And let's wipe that off, Bill, though it would actually take a minute or more. And then what we do is compare that with the color scale on the packet, and we see whether we can read from that how much glucose there is in the blood, measured in milligrams per 100 milliliters. Well, that's a quick and straightforward way of doing that important test, which depends again on enzymes and which diabetics increasingly will be using. That's the sort of way in which knowledge of how enzymes work is affecting the practice of medicine and all sorts of other things. I hope I've given you some impression of how exciting a subject that is at the moment. It is, I think, about the most exciting time there's been in molecular biology, even though people thought a little while ago that it could never possibly be as exciting again as it was back in 1953 at the time of DNA. So let me end with just two quick examples of what the present excitement is about. The next slide shows a person who is very much responsible for a lot of the information I've been showing you. This is Fred Sanger, who was the first person to find out the order in which the components are linked together in a protein chain. It was the chain of insulin. And for that, he had the Nobel Prize in 1958. A few years ago, he devised a method for working out the order in which the units are linked together in a chain of DNA. And for that, he had the Nobel Prize for the second time, just last year. Now, what he's been able to do is to look at the sequences, the arrangements of these bases in DNA. And here's a paper in which his first results were first published in Nature. You see long, long lists of letters, A, G, T, and C there, which tells us in great detail about the way in which the genes are organized. A lot of new information is coming out of that, and I'll just give you a hint of some of it. You may have wondered when we were talking about protein synthesis here, where we were reading this message in three-letter words. I better move it so that we can see two three-letter words. What would happen if we moved on one letter? Well, it's clear what would happen. Instead of reading GGG, we would read GGU, and although I don't remember, that may well be a different protein component. So it's important to read starting at the right place. A thing which Fred Sanger and his colleagues found in this piece of DNA, which comes from a bacterial virus, is that the virus packs information into that very tightly, and in this little piece of the sequence, it's only a tiny little bit. We can see some of the letters there. This sequence, if I read it up at the top, from this point, reads GCU, AAA, GAA, UGG, AAC, and that means A, K, E, W, N. But the virus also arranges to read it starting there, and that means M followed by E, and so on. It's coding for two proteins in the same bit of DNA. That's the sort of information that's just coming out about the structure of viral genes, and equally exciting information about the structure of our genes. What's the other bit of work? Well, I can introduce that with a rather old piece of work done by John Gurdon back in the 1960s. This has to do with cloning, if you like, how to produce identical copies of creatures. What he did was to start with frog spawn. There's an egg. Irradiate it with ultraviolet light to destroy the nucleus. And then, having taken a cell from the intestine of a tadpole, there are the cells, he took the nucleus of one of those cells and injected it into the frog spawn egg. What happened? In 1% of the cases, a very small proportion, the egg with a new nucleus grew up to be first a tadpole and then a frog, this sort of frog. It's really a xenopus toad, just like the one related to the tadpole from which the cell was taken. So cloning frogs was possible. That work goes on, and it goes on not for fun, but to get new knowledge. That knowledge we have to use responsibly as citizens, and I hope that you will play a part in getting it. Cloning human beings may eventually become possible. We don't know enough yet to know. But we do know, I think, that if you see it in the near future, you can be sure it's done by clever photography and clever electronics. And however it all turns out, I hope that you will agree with me that the work must go on, and that ignorance and prejudice must give way to knowledge.